Yeah, thanks for the introduction. Can you hear me now?
Yes.
Okay. So hello, everybody. My name is RyoMizuuchi from the University of Tokyo, Japan. And I would like to first thank organizers for inviting me. It's a great honor to be here as a speaker. And today I'm going to talk about evolutionary complexification of a replicase encoding RNA replicator, which is extended works from what was presented in the keynote talk. Sorry. Okay. So our question is, can a replicating genetic molecule complexify through evolution, which I believe is a fundamental step towards the emergence of a complex life? And this quest of evolution perhaps started from an RNA replicator, possibly in combination with peptides, proteins, and many other biological molecules. And I think we all agree that they should have expanded continuously their information to encode diverse biological information, biological functions. And we are focusing on, we are trying to understand the very first few steps of this evolution. And historically, this has been studied theoretically in detail, and the likely scenario has been proposed. I'm not going to very detail, but basically from an RNA, the new RNAs successively emerged by evolution with new information and functions. And at this stage, they replicate as a network by cooperating with each other so that increased amount of information can be stored in the whole system. And then they got integrated into a single long chain, which is reminiscent of contemporary genome that encodes diverse biological information, and thereby completing a complexification step from a smaller unit to a larger unit. One analogy of this evolution is evolution of multicellularity from a unicellular organism, and a similar evolution may have happened in the origins of life as well. And although many theoretical studies supported this idea, no experimental evidence was available. And that's why we try to observe it directly by experimental evolution. And today I'm going to first talk about this network formation part, and then I'm going to also talk about the unification using the experimental evolution system. So what kind of system are we using, which was described by Ichihashi-san, so I'm just briefly describe it. We are basically using an RNA protein replication system, which was probably not available on the early Earth, what we believe it mimics primitive replicator in a sense that it has only minimized information and functions. So our system consists of an RNA encoding qubit phase replicase, RNA polymerase, and the translation system derived from E. coli and encapsulates in water and oil emulsion. In the compartment, an RNA replicates through the translation of the self-encoded replicase. And the compartment makes sure the replicase can replicate the RNA from which the replicase is translated. And during the replication, mutations are introduced, and the variant RNAs that could have different characteristics would be generated. So using this system, we performed serial transfer experiment, evolution experiment. We basically incubated the RNA replication system at 37 degrees for 5 hours for replication. Then 5-fold diluted the population. Then we supplied the nutrients and translation system through fusion division compartments. And as Ichihashi-san introduced, previous studies performed 120 rounds of this evolution experiment and found that during replication, sometimes causes deletion to generate a short plastic RNA that cannot replicate by itself because it lacks the replication gene, but it can replicate by utilizing the replicases translated from other RNAs. And we call replicase encoding RNA as a host because it's parasitized. And parasites usually replicate faster because it's short. And these two RNAs co-evolved through adaptation and counter-adaptation. And to some extent of diversification was also observed. And such a great ability of adaptation made us think that more evolution may lead to further diversification and then eventually the formation of a complex replication network. And thus, this time we continued the evolution experiment and performed additional 120 rounds, in total 240 rounds. So this is the replication dynamics. X-axis round, Y-axis is RNA concentration. A blue represents host, and orange, green, purple represents parasites of different length. Oscillation is notable, and that's because at high concentration stage, many parasites replicate and inhibit host RNA replication. Once reaching, and then they are diluted, and once reaching low concentration stage, the number of molecules in the compartment decreases, and the host RNA replicate again without severe inhibition. And in this graph, we didn't care about variations in each species. And that's why it looks there are only two surviving species, blue host and the purple parasite here. And so next we performed next generation sequencing to analyze these two surviving species in detail and fully understand what happens and whether they complexified. And so here's the outcome shown as a phylogenetic tree, and we basically found both hosts and the parasites diversified significantly. And the heat map in the inset shows the frequencies of each RNA over rounds from left to right, and the list of dominant mutations were also displayed here. So it started from the ancestral RNA here in host lineage 0 or HL0, it evolved and first diverged into green branches and blue branches, part of which was already described by Chess-san. And we defined green branches as a single lineage, HL1, host lineage 1, because the heat map shows evolution in one direction as well as proceeded. And the black stars indicate RNAs that remained in the final round. But on the other hand, blue branches showed a much more complicated dynamics, which we defined HL2, and it first diverged into parasites in purple here, although they quickly went extinct. Some RNAs in HL2 accumulated, some RNAs in HL2 remained in the final round without accumulating many mutations indicated by the short horizontal branches. But other RNAs accumulated independent set of mutations and make another set of branches in navy, HL3. And moreover, HL2 diverged into new parasite here in pink, PL2, and they also diverged into magenta parasites here. And the HL1, 2, 3, and the PL2 and the 3 remained in the final round, accumulated different sets of mutations. And as I'll show you later, they show distinct biochemical characteristics. And next, we analyze the frequencies of each lineage over rounds, x-axis round, y-axis frequencies, hosts at the top, parasites at the bottom, and the horizontal bars above the graph indicates where each lineage was detected. So for example, looking at the host lineages, they were detected at some rounds, but undetected in other rounds. So their frequencies were highly fluctuated in the early stage of evolution, like this. But in the last 50 rounds or so, 3 lineages, HL1, 2, 3, green, blue, and navy, were constantly detected at roughly more than 10% of the population. And similarly, 2 parasitical lineages, PL2 and 3, were also constantly detected at relatively high frequencies in the last 50 rounds or so. So basically, both host and the parasites diversified and underwent transition into a quasi-stable coexistence of multiple lineages. And the transition to the coexistence, and we thought such a transition may be associated with the formation of some kind of interdependent replication network. Then we next analyze the relationship between dominant host RNAs and the parasitic RNAs at these 4 indicated rounds. Which RNA can replicate which RNA? Sorry. And here I show the relationship as direct graphs. Basically, a node or circle represents an RNA, and edge or arrow represents replication, and its width corresponds to the extent of replication. So started from a single ancestor, the both nodes and edges were gradually increased and finally evolved into a 5-member network. And in this network, among host RNAs, HL1 replicates itself, but also replicates PL3. HL2 not only replicates itself, it also replicates any other members in the network. So it's highly cooperative. And the emergence of such cooperation is particularly important because the cooperation can unite distinct components to make a higher order system. HL3 is mainly replicated by HL2, but it also replicates PL3. And among the parasitic RNAs, PL2 can be replicated only by HL2, but efficiently. On the other hand, PL3 can be replicated by any host RNAs, but modestly. So all 5 members show distinct replication strategies. And I will stop this topic here, but our results also suggest that these RNAs sustain each other's replication because removing any species from the network disrupted, including parasites, disrupted the whole system, at least in simulation. And many more analysis can be found in the preprint if you're interested. So now I showed RNA replicator can evolve into some kind of network. And so the next question is, can they get integrated into a single long chain? And that's I will quickly introduce. And for this investigation, we use a different replication system constructed previously. So there are 2 RNAs, one encoding replicates, the same replicates, and another RNA encoding NDK, nucleotide diphosphate kinase, a metabolic enzyme. So in this system, in a compartment, NDK is translated from one of the RNAs and it converts CDP into CTP. And replicase translates from the other RNA, replicates both RNAs using the synthesizer CTP. So these 2 RNAs are obligatorily mutualistic. And in this system, very rarely, these 2 RNA fragments got integrated into a fused version of the RNA. We call it fused RNA by replicase associated or spontaneous recombination ligation, which I'm actually working on in another project. And that's why they are parasites. And we wondered whether this fused RNA can be selected in the serial transfer. And we first simulated the serial transfer and found that started to form only fragments in the presence of parasites, and thus through oscillation, this generated fused RNA can be stochastically enriched. And this is partly because fused RNA has advantage over fragments in low concentration stages because both genes can be encapsulated in the same compartment at the same ratio always. And that's a reason why we chose this obligatorily mutualistic replication system. And we are now testing this in actual serial transfer experiments, mainly performed by Kensuke. X-axis is round, y-axis is RNA cancellation. Oscillation happens because of the parasite, partly because of the parasites, which are not shown here. And this cancellation represents each gene region, so which may represent fragments as well as fused RNAs if they already emerged. And next, we did fused RNA specific RT-PCR, 2 possible conjugations, blue fragment followed by yellow or yellow followed by blue. And we detected one of the 2 bands at around 36 and both bands at around 79. So fused RNAs may have already emerged. We next sequenced the bands and confirmed they were actually fused RNAs, A and B. And the next we identified dominant mutations and compared them with the product of each gene region, C, replicase, or D, NDK, and these may represent the fragments or part of fused RNAs. And we found that the mutation pattern of a fused RNA such as A is quite different from the pattern of fragments such as C. And this result indicates two things. First, fused RNAs have not dominated the population yet, because if so, the mutation patterns should be the same, such as A plus B equals C plus D. And second, fused RNAs have unique evolutionary histories. So which means fused RNA probably replicated at least for some rounds. And we are now continuing this evolution experiment to see if the fused RNA could dominate the population eventually. And now I'm coming to the summary. So we evolved an RNA protein replication system. And we basically observed spontaneous network foundation from a single RNA replicator to a multiple replicator network. And we also observed the unification of the constituent RNAs in a different replication network. So our conclusion is molecular replicator can complexify, and more serial transfer experiments, we believe, would offer more insights into a possible evolutionary pathway that may have led to the emergence of living systems. Then I thank my co-authors, Norikazu Ichihashi, Taro Furubayashi, and Kensuke Ueda, and funding sources. And thank you very much.